Hi, my name's David and I'm a biology teacher here at the Dublin Academy of Education. Today on Whiteboard Wednesday, we're going to cover photosynthesis, which is worth between 6 and 15% of your Leaving Cert exam. It has come up every single year, except for 1 year in 2014. The two questions that I feel cover the entire topic are question 14A from 2013 and question 12A and part B from 2015. And if you get a good grasp of that, photosynthesis will be absolutely no problem to you. What we're going to do here is look at a little bit of an introduction and how that photosynthesis is a plant using light to produce food. We need to know this equation up here, which is pretty much the exact same, but back to front of the respiration one we have learned. So we've 6 carbon dioxides plus 6 waters in the presence of light, gives us glucose and oxygen. Some things that students just need to be aware of is the difference between a chloroplast and a chlorophyll. Chloroplast is the actual place where photosynthesis occurs, and chlorophyll is the pigment that traps light that actually fills the chloroplast. So don't get those two mixed up. Plants are a term called autotrophic. We are heterotrophic, and autotrophic means that it can actually produce its own food. And what does this do? What does the food actually do? The fact that a plant can photosynthesize do for us? Well, it produces oxygen, both for itself, for respiration and release into the environment. It produces plant food, and then either through itself or then actually producing other things can produce animal food. So there's 3 functions. We need just a basic introduction to it, and I've put it down into 5 steps because this is an examiner's favourite, but we don't need to go into massive detail. So step 1, light gets absorbed by the chlorophyll, the light predominantly coming from the sun. Step 2, water is split, a process known as photolysis, so splitting using light. Step 3, that split forms 3 products from water. So it forms electrons, protons and oxygen. Then what happens is the excess light that goes in can energize these electrons. They go off and do certain things, and that energy actually then produces glucose using carbon dioxide. For part B, what we are going to look at is the actual processes of photosynthesis in a little bit more detail. I'm going to point out to you the ideas that I feel that you should really be aware of for this year's exam. So first of all, it's split up into 2 different stages. We have the light stage where light has to be present, and the dark stage or the light independent stage where it doesn't. In the light stage, which is the first part of it, light gets absorbed and then it actually produces a few products, and then the light gets transferred to electrons. So the energy from the light goes to different electrons. This is the part that students find very difficult, the 2 separate pathways. We have pathway 1 of these electrons, which is a cyclic pathway, meaning it just goes round in a circle and starts again and again. These high energized electrons bounce around, lose some of their energy, which gets trapped and forms ATP, which we've seen before. Then they go back to the chlorophyll, and again they just repeat this process again and again. Pathway 2 is non-cyclic. These electrons do not return. So photolysis occurs, which we've talked about before, producing protons, electrons and some oxygen. ATP is produced exactly like before. The electrons lose some energy and produce this ATP, but in this case they definitely do not return. That's really important. That's what makes it non-cyclic. And the second part of this that students find a little bit difficult is these electrons then join on to NADP for photosynthesis plus, along with a proton to form NADPH. The final idea here is there are 3 products that come from this. 1, ATP, 2, this NADPH, and 3, the oxygen, which we know gets released into the atmosphere and is also used by the plant for respiration. So these 3 products then go into the second stage, also known as the dark stage, where light is not needed. So also it can be called, and a common question is, what's the alternate name? The light independent stage. On older past paper questions, you might see it referred to as the Calvin or the Melvin cycle, but don't worry too much about that. This stage is controlled by enzymes, and we know enzymes are actually affected by pH and temperature. So it's really important that the plant has an optimum pH and temperature for this part to work. Again, this occurs inside the chloroplast, exactly like the rest of these stages here. So not in the chlorophyll, inside the chloroplast, just a different part, but we don't have to be too worried about that. And what happens is carbon dioxide from the atmosphere is absorbed. It then combines with the products of the NADPH. Remember what that is carrying? That is carrying the electrons and protons from before. And it forms glucose, which we know to be C6H12O6. So you can kind of see how that occurred, how it joins into that. The energy needed for that comes from ATP, just like before. So for the experiment that we need to know in photosynthesis, we're going to concentrate on the light intensity one. You can either do this or the one where we change carbon dioxide, but most students would actually do this. You do not have to do both. So we're going to see how different light intensities affect the rate of photosynthesis. We have a setup like this, which includes a pondweed Elodea. We'll talk about that a bit more in a second. We have a little container here, a water bath, keeping the temperature the same, we add some pH buffer to keep the pH the same, and we add excess sodium bicarbonate, so there's enough carbon dioxide for photosynthesis to occur. What we then do is we place a light beside the apparatus, and what happens is the Elodea starts to photosynthesize. And we can see this from bubbles rising up through the tube and collecting at the top as oxygen. What we want to do though is see how, when we affect the light, when it gets stronger or weaker, how this affects the rate of bubbles. So we have to put it in 3 or 5 or how many places you put it in school, leave it settle, and count the amount of bubbles per minute or per 30 seconds. We can then draw a graph of our results. Now in the leave insert we don't need to have any numbers or anything like that, but it looks a little bit like this. The closer the light gets to the actual apparatus or to the pondweed, the rate of photosynthesis goes up until it hits a point about here. And this is known as the saturation point. That's the absolute maximum amount of light that the plant can absorb. 2 other things we need to be really aware of, because the examiner likes to ask this, is why do you use a pondweed? We've studied like nothing to do with pondweeds. Well, this actually allows us to count the bubbles underneath the water and give it a rate. If we had a plant just out in the atmosphere, it would be impossible to do so. The second bullet point here that we have to be aware of is you have to definitely leave this light to stabilize for a minute or 2. So you can't just start counting the bubbles straight away. We got to put it down, leave it for a few minutes, and then start counting it before we move it again. So just to recap, photosynthesis, 6 to 15% of your paper every year, bar 2014. It can also help us in a few other questions. We've broken it down into an introduction and a basic idea of it, which is an examiner's favourite question. The detailed part, I've starred here that I feel that pathway 1 and 2, explaining them and what we have on the board is going to be quite important, not in too much detail. And then the experiment over here, which we definitely need to know inside out. Thanks very much for watching this video. Check back next week for a different topic on Whiteboard Wednesday.